Hello and welcome. This is Columbia Basin Research's fourth video in a multi-part series on using Program Atlas, which stands for Active Tag Life Adjusted Survival. Atlas is a program created by Columbia Basin Research at the University of Washington's School of Aquatic and Fisheries Sciences with funding provided by the Bonneville Power Administration and the U.S. Army Corps of Engineers. Today I'm going to show you how to load in and work through a single release. If you have not run through lessons 1 through 3 detailing the ins and outs of Program Atlas, working with capture histories, and working with tag life data, we highly recommend you watch those before going through this video. First, let's fire up the program. Type Atlas or shortcut to Atlas into the search bar, then select a single release study from the front end menu. If you were already working within Atlas, you go to Edit here, change study design, and select single release and hit OK. A single release study consists of a single release group with multiple downstream detections. It may be either an actual single release study with a release group consisting of a single release or a virtual single release study with a single virtual release comprised of fish from multiple upstream releases. When I load in the capture history file in single release mode, selecting Load Capture History, and my single release data file, this dialog box appears, allowing me to select the type of single release study. Upon selecting the single release study type and then pressing Next button, the user must next select which releases in the dataset to include in the analysis. In the case of an actual single release study, only one group can be selected, so I don't have to click anything. If a virtual single release study was selected, one or more release groups can be selected to form the virtual release. The review box comes up detailing my choices. I can click Finish if content and back to revise any previous choices. Since my actual release only contains one group, I can't do much with define release groups, or anything at all with view mixing. I can, however, check or uncheck the last two downstream sites. The single release allows me to view the capture history report, CJS report, cumulative CJS report, and the CJS covariance report. Further information on these reports can be found in Lesson 2 on working with your capture history data. I'm going to skip attributes again. We'll go over those in the final video in this series. I can now load in my tag life data. Double-click Load Tag Life Data to load in your tag life data file. Then double-click on Fit Tag Life Curves. You can sort through the various functions to see which one best fits your data. The Weibull 2 and the Weibull 3 curves don't fit very well. The vitality curve fits best in these circumstances. Click Apply. From here, we can look at curve versus data by detection. We can see that in this particular scenario, not all fish arrived before tag life began to fail. I don't have different lots in this session as I'm just working with one single release capture history and one tag life history, so we can skip map tag life data to lots. I can now double click on Calculate Tag Correction Table, enter the number of bootstrap iterations, 1000 recommended, and wait for the calculation to proceed. It can take a few minutes. I won't make you wait it out here with me in this video. Now I can look at my tag life correction table, closed form parameter report, and cumulative closed form survival report. Under modeling survival estimates, I can't equate parameters across releases because there was just a single release. I can, however, constrain parameters. Since a 1 is considered infinity in the next bootstrapping process, I have to ensure I check any 1s found here. Here's one under capture. Not doing this will cause the next computation to error out. Click Apply. I can now compute my results and estimate full variance and look over my parameter report, cumulative survival report, and survival covariance report. Going through these steps if you were running a virtual single release study design would be quite similar, the difference being that the first site now defines a virtual site. The tag corrections table must be interpreted differently than in the case of an actual single release study. The tag corrections column, D0, are the unconditional tag survival probabilities from the release site to D0. Under the subsequent columns, D1 through D3 in this case, are the conditional tag survival probabilities given that the tags were alive at site D0. Note the tag corrections under D0 are not used directly in the calculation of the corrected survival estimates. They are used to calculate the subsequent downstream tag corrections and are displayed in the tag correction table for reference. This has been Lesson 4 on using Program Atlas, in which we worked our way through a single release and the differences between actual and virtual single releases. Future videos in this series will walk you through paired and virtual paired releases and attributes, as well as offering some hints and tips on maximizing your Program Atlas experience. We hope that this video helps you get the most out of Program Atlas. Again, if you have any questions or comments in regard to Program Atlas, please fill out our survey as previously detailed. You can also contact Columbia Basin Research at web@cbr.washington.edu. Best of luck with your analyses, and thank you.